know if it's in vesicles or it's packaged somewhere else, but it does seem to be there.
Right, but I'll also say another limitation of this study is that the sampling time is 12 and a half minutes. So we're collecting a sample
Mm,
every 12 and a half minutes. And if you're thinking about an intense cardiac arrest event, it's probably going to happen on the scale of seconds or less.
I see.
And if we're grouping all that into a 12 and a half minute sample, then we're likely missing a lot of the important nuance of that process.
Hmm. So based on that evidence, would you say it shows that there is endogenous DMT in the brain under baseline conditions? Or would you what are the conclusions you would actually draw from that study that you think are rock solid?
I think that's the main conclusion. Yeah. And that was actually the first study to show that DMT is present in the living rodent brain, freely behaving rodent brain. A lot of the older studies that had showed DMT in the brain, it's like postmortem tissues.
Hmm.
So which is still valid, but this is sort of showing that DMT is present in the freely behaving rodent brain. So I think that's probably the most solid takeaway from that study.
Hmm. And so I guess the a question at this point is the DMT there because it's only to protect the brain from hypoxia or something like that under special conditions? Is it some kind of, stress protection type molecule? Or, the more provocative, the more interesting hypothesis would be, no, it's actually used as something like a neurotransmitter. And so, unpack that for us, like what the thinking is there, what the hypotheses were, and how that feeds into the work that you did.
Yeah, so I think it would need a lot more evidence to be characterized as a neurotransmitter. The hypoxia hypothesis is a very interesting one, and it seems there's some data out there to suggest that it is functioning in a hypoxia protective role. But in order to characterize a molecule as a neurotransmitter, there's a number of criteria that have to be met. And if you look at some of the canonical neurotransmitters like serotonin, you'll see that there's specialized neurons for their synthesis. So we have serotonergic neurons, and those neurons have the enzymes necessary for the biosynthesis of serotonin. So we would need that for DMT. We would need DMT-ergic neurons, which there may be. Another finding of that study by John Dean is the co-localization of those two enzymes, INMT and AADC, was found in a number of different brain regions I think in the cortex, in the choroid plexus, and in the pineal gland as well. So those could be areas where there are quote unquote DMT-ergic neurons. So that's one criteria for a neurotransmitter. Another criteria is that there's a mechanism to package and store the neurotransmitter in vesicles. That's generally done to protect the neurotransmitter from metabolic degradation and to sort of store and sequester it so that it can be released via exocytosis in a concentrated package. Another criteria is that once that vesicle is released via exocytosis, that the neurotransmitter activates a postsynaptic receptor. There's some receptor on the other side of that presynaptic neuron that gets activated and causes a downstream physiological response. Another criteria is that after that postsynaptic receptor has been activated, the molecule would need to be recycled or metabolized in some way, and often that's a reuptake mechanism. Like for serotonin, it's SERT, it's the serotonin transporter that can take serotonin back into that serotonergic neuron, and then either metabolize it or package it back into a vesicle. And that exocytosis has to be activity dependent. There has to be some physiological response that's causing that exocytosis. So those characteristics still need to be worked out if we're going to start to call DMT a neurotransmitter or a neurosignaling molecule even. And there's a bit of evidence kind of for each of those. So you could very loosely kind of put together a hypothesis that suggests that DMT is a neurotransmitter, but it's not quite solid. And a lot of it's from pre-1980. So there hasn't been much modern investigation into
Mhm.
it.
So is a fair summary. So all those criteria need to be met before you can say something is a bona fide neurotransmitter in an animal brain. We know that there's some DMT there, We don't know how it's packaged. Is it in a synaptic vesicle? Is it packaged some other way? We know that obviously DMT can activate postsynaptic receptors, right? Like there's 5-HT2A receptors in the brain. If it is being packaged like a neurotransmitter, which no one has proven yet, there would be receptors there for it to activate. But we don't have those other pieces, right? We don't know if it's in vesicles. We don't know if there's neuronal activity dependent release.